Maybe you could, maybe you could.
Okay.
Yeah, no, that, we'll think about something else.
So you all know that you have borrow. Keeper dam. Old school. Keeper dam. Anyone have any questions on that?
What would you want us to know about, like, identifying companies? Just because on here, be able to describe a company.
Colony, yeah, so, you know, when you look at street plates or your spread plate results, when you're looking at those colonies, like, what should you be paying attention to? Like, what is it about the colony? What kind of information can we get off of it, right? So if you're looking at, one of the things that we do is you look at the perimeter or the circumference of the colony, you look at the color, is it transparent or opaque? We're not asking you to memorize all those fancy terms that are in the PowerPoints. I prefer you kind of describe things in your own words and you take notes in the lab so that you can remember it. So, like you're going out to the search lab microbiologist, but you do need to understand that colonies can give you a wealth of information. Once you train your eye and start getting more experience at it, it can really give you a lot of information. That's what we're trying to get you to think about. Other questions? Isabella was sick, right? She didn't make it into the lab yesterday. You know, before the night before, so contact took over. So, but if Isabella's not back by tomorrow, we will talk to her myself. We'll check in. Talk to her a lot. Okay. So we are on the road with endospores. We've been talking about that. And so gram positives so far are the only type that we're aware of that can do this. We talked a little bit about how it's a big, long process. Once they start to engage into it, they're not going to back out of it, even if conditions really get much better for them. So basically, these bacteria that can do this, they're constantly assessing their environment like all other microbes. It's just that they have this extra option. So if times get really tough for them, they can decide, you know what, I think I better go into endospore formation. And at least that will allow us to survive as a species moving forward. And so it's all within the vegetative or mother cell. And it's pretty complicated. It's multiple layers of protein and so the exosporium and spore coats. It's just a lot of layers of very special protein. And then you finally start to get into where you start to see the a little bit of peptidoglycan, and then right under that, you start to get to see the proper, like, full cell wall of the microbe, the cell membrane, all the inner components. So they build all of that inside the vegetative cell, and then the last step that has to happen is everything's got to dehydrate pretty dramatically, so that if it stays too moist, it'll decay or get broken down or attacked by fungi in particular. So where did we go with? Where did it go? So here, this idea that you have special chemicals that can help with the stabilization of the inner cell deep in the middle of the endospore, so that as it's drying out and dehydrating and getting ready to go dormant, it can stay good to go once times get better for the microbe, which is usually, you know, well, at least in the lab, one of the things that we do is if you dehydrate it, you know, just withdraw all the water. Then if you supply water back, that usually gets them to germinate. So were you able to see endospores with the endospore stain? Were some of them in the cell still? Okay, good. It's really tricky to time how much to grow those bacillus species for you to make sure. We want you to see spores in the vegetative cell and also some floating out. And so, but that can be tricky to do. So hopefully it worked well. And then I did put this I tracked down, I remember, where did it go now? Here. I was trying to come up with hard numbers and I couldn't remember the other time we talked, but this is from a table taken from a microbial ecology textbook. And ST stands for survival time. So you see here in a laboratory test tube, they let Clostridium endospores just sit in a test tube for 34 years. And then they sampled it and then it was still fine. And then here you can see spore forming bacillus species in the gut of an extinct bee that was trapped in amber. So they're saying it was roughly dated, that amber piece was roughly dated to 40 times 10 to the 6 years ago. So that's 40 times 6 zeros. That's a lot of time ago. And then here, 100 times 10 to the 6 years for if you want to go deep into clay and shale. So look, these are often microbes that are way deep in the ocean, earth crust. But here's another endospore, 250 times 10 to the 6 years ago. So really impressive endospore. I mean, their abilities to withstand that and stay dormant that long, and then they were able to revive them all these years later. It's pretty amazing. So endospores, they are amazing. They slowed my research down by about a half a year. So like I said, I have a love-hate relationship with them when they got contaminated in the various things that I was working with in the lab. I think we left off maybe around here, just showing this idea of once the spores sense, it's time to start to germinate, they will grow out of the endospore and essentially, result it back to the vegetative state. It'll look just like the original vegetative cell. Okay. Couple of other things I wanted to mention is that sometimes when you look at microbes under the microscope, especially environmental samples, you might see these weird little things inside the cell. And often they'll like bounce light back out. So it's like this bright little white spot. And what you're seeing there, in this case, those are sulfur granules. And so microbes, when they're surrounded by really, feast conditions and they're getting well fed, they got everything they need. Sometimes if they need, or if that's the situation, they can actually store up certain nutrients inside their cells. Now they're tiny, so they can't do a ton of this, but here you're seeing pretty well-packed cells, lots of sulfur granules. So then in times of famine, they can start breaking down and using that sulfur for building whatever. Some certain amino acids need sulfur and other nutrients. And so you can have that. You might have phosphates that are stuck together, phosphate groups that are stuck together. So they're polyphosphates that will get stored in some cells, even glycogen, which is our storage form of carbohydrate. Some microbes can do that too. And then here on the right, you're seeing pictures of big oil droplets, lipids. And so again, some microbes under feast conditions, they can store up certain nutrients and wait, as times get tough, they can start using that and breaking it down. And then I think your book still mentions this. I was surprised to learn when I was a grad student in microbial ecology classes that there's magnetic bacteria out there, and they contain magnetite. They are usually ocean dwelling. They find that they're suspecting they're using it for navigation in the deep, vast oceans, like a compass. So it's still kind of mysterious. I didn't know this until somebody pointed out that studies animal phys, that humans, we also have magnetite in our brain. Does anyone learn that? I didn't know that till I was teaching here. So I don't know what that's about, but anyways, I don't know what magnetite does for us. If that's a holdover from billions of years ago or not. If anyone figures that out or lets me know what somebody might have said about that, let me know, because I just find that really bizarre. All right. So that is the end of the cell structure and function material that I wanted to go over. Is the next one posted? Ooh, let me post it. I think it's because I meant to update a few things, so I may end up updating this a little bit because there's a couple of things that I tweaked. Let me go into Educat and get that. Okay, so it's visible now. Intro to metabolism. Let's do a Google form. So this idea of anabolism is like anabolic steroids, where you build up your muscles if you're on steroids, that type of idea. That's the idea of, building more complex polymers from simple molecules. Whereas catabolism is the breakdown. And the way I remembered that for catabolism, when I was an undergrad, we had a family cat that would climb the living room curtains, and he had full claws, and you would shred the curtains. And so I think about shredding complex molecules, catabolism. That's how I remembered that term. So little tips and tricks like that you can figure out to play with the words so it makes sense with your brain is a good idea. These two are linked. I said they're two sides of a coin, but they're definitely linked in the idea that metabolism does provide the energy needed for anabolism. And you don't have 100% conversion. There's some losses heat. Nothing's 100% efficient. So just like our bodies give off heat as well as we're doing this. So here you have the idea where you're going, a lot of what's being shown on here is kind of the anabolism idea, where you're taking simple molecules, putting them together to more complex molecules, and to make sure that you have a intact viable cell cooking along, doing its thing. I also put this picture on here too to remind you that even though we can't see these microbes, they are busy doing a ton of stuff. And a lot of that has to do with, I need ATP. If I don't have it, I'm gonna die, and die quickly. And so we need to think about that. Also, a little shout out to your microbes in your gut. You have some fascinating interactions going on. And one thing I want to emphasize is that many situations, when you think about what you eat and then it gets into your intestinal tract, you typically have a whole group of microbes working on that food. You don't just have one microbe feasting and all by itself. You typically have microbes that are hanging out in close proximity. They're all doing some of their own stuff, but then they also team up to help break down some of the complex. Hopefully you're getting some complex fiber in your diet, right? Fiber's really good for you. I'll talk more about fiber as we get into human microbe. And so here, what they're showing is this idea of, and we'll talk about this too, extracellular vesicles. So microbes are known to like balloon off part of their cell membrane, and they can enclose certain, in this case, enzymes that break down your food that you've eaten, and they'll put that out into the nearby environment for other organisms to share. And so they have this many strategies to work together as a team, because some microbes might have a particular enzyme to break down a portion of the food you eat, but they can't do it all. And so then the next microbe nearby says, well, I have this enzyme and I'll break down this part. And so it's always this teamwork out in nature too. It's not just in your gut. Natural environments are like this too, where you don't have microbes living in isolation. You have them working together. And that's why when you start thinking about biocontrol ideas of plant diseases or bioremediation of toxic environments, you often have to think about a consortium or a mixture of microbes so that they can get the job done correctly and hopefully do what you want them to do. So I'll come back to this idea when we get into the human microbe, but it's basically trying to show how these cells, I think it's a starch particle, So they've attached to the starch particle and they're starting to break it down, and then they're also like releasing and sharing some enzymes that they have out for others nearby to work with. All right. So to get usable energy, ATP, you got to go through catabolic reactions. I believe some of you in here, when I looked at all of the different majors, is quite diverse this semester in terms of all of the different majors that you represent. But there are some biochem majors in here, phys majors, and then if you've taken some basic, maybe intro to cell or even a little bit of BI 111, you might have covered the basics of glycolysis and then the TCA cycle, also called the Krebs cycle or the citric acid cycle. And then the so-called electron transport chain process that happens in your mitochondria, often referred to as aerobic respiration. So we are all currently doing this right now. Hopefully you've had a decent lunch and your cells are busy doing this. And so what I'd like to do is start off going over these and giving some of the big picture concepts. This is not a biochemistry class, so I'm not going to ask you to know it step by step. But as we work through this, I'll point out what I want you to know. So what I like to do is start with this because at least you have, I'm guessing most people in here have some vague familiarity with this. And then I want to build off of that because you're going to get exposed to stuff probably you've never heard about. But hopefully this will provide some foundational knowledge for you. Because we're going to find that, yes, microbes can do all this too, but they have some other strategies in play that we just can't do. Microbes are incredibly versatile when it comes to their physiology. I took a whole year of microbial physiology classes as a grad student. I could have easily done a whole PhD just on that. It's very complex. So if it's been a while since you've thought about ATP, I'm seeing that here with the 3-triphosphate idea. So I remember learning about that when I was an undergrad. I never remembered learning about this when I was an undergrad. I'm going to blow that up into the next slide. Did you know that ATP has to have magnesium in order to function? I didn't know that. So this is a quote from the book. Under physiological conditions, ATP always forms a complex with magnesium. It partly neutralizes the negative charges of the phosphates, stabilizing the structure. In solution, most enzyme binding sites for ATP actually bind magnesium ATP. This is one reason why magnesium is an essential nutrient for all living cells. I don't know if you've noticed, but out in the general media, I see, well, I don't know, maybe it's, what do they call those? The algorithms on your phone. But I'm seeing a lot of magnesium, like supplements, probably because I did a search for them and now it's showing up on my phone all the time. But it's a big money. It's a big industry out there selling magnesium to humans, which let me just say, some magnesium is better than other types of magnesium in terms of how bioavailable it is to your body. So just be very careful about if you decide to buy magnesium, you have to buy the right kind. I'm happy to talk to anyone. I've done a deep dive on that because I take magnesium. But anyways, there's all kinds out there and some don't work. But I had no idea. ATP has to have magnesium. Okay, so we need to build ATP. These microbes are doing it. So I wanted to start with glycolysis. Technically, glycolysis is a fermentation reaction. And if you, I think your book phrases it this way, it's a fermented substance that's both an electron donor and an electron acceptor, is how they often talk about it. It's anaerobic because there's no oxygen involved anywhere in the line of the process. So basically it's a type of fermentation, and we tend to break it down into 3 stages when we're learning about it. Now, when I took biochem, I had to learn every single step, every single enzyme for every step. We're not doing that, all right? So let me just show you some overview statements. Stage 1 is what they call the preparatory reactions. And that's where we usually start with glucose as we talk about glycolysis. We bring in 1 glucose into this pathway. By the end of stage 1, you'll have glyceraldehyde, 2 molecules of glyceraldehyde 3-phosphate. So remember, glucose is 6 carbons, G3P are each 3 carbons. All right, so you get 2 G3P, glyceraldehyde 3-phosphate. I think it'll be spelled out on the figure that I show you. It actually, you have to use ATP to get this stage done. You want to get it to completion and done correctly, you do have to cough up some ATP. So they often refer to this as priming the pump. You got to get things, a little bit of a nudge, and then after that, you'll get some payoff. All right, let's go to this figure. This might be slightly different than the one that's been updated in your book, but I like this. It's a little cleaner to my eye. So what do we have? We have glucose coming in, and they've color coded it into yellow, that by the time you're all done, you're going to be right there, glyceraldehyde 3-phosphate. You have 2 of those after 1 glucose comes in. You can see that the ATP is being used. I don't expect you to know where it's being used, just know 2 ATP gets used in stage 1, and you start with glucose and you end up with G3P. Then the kind of, I guess, pink section now that we're moving into, that's stage 2. So let's just think about that broadly, what's going on. Stage 2, the goal is to make pyruvate and ATP. So that one of the questions I asked you about what's the product of glycolysis when you think about animal or human fizz, it's pyruvate. So basically you're shifting from glyceraldehyde 3-phosphate, going through a series of reactions, and you end up with pyruvate. In this stretch during stage 2, you do get 4 ATP. So then you get a net total of 2 ATP. By the time glycolysis is done to this point, you used up 2, you made 4, you're left over with 2. So you get 2 ATP going from glycolysis to pyruvate. It's not a ton. That's a lot of work for 2 ATP. So that has some serious ramifications for microbes who can only do this type of fermentation. So the fancy word for what's happening here, when you end up with making some ATP through this method, this is an example of what we call substrate level phosphorylation. So when you look at this phrase, what do you think they're referring to when they say phosphorylation? What's getting phosphorylated that we are keying in on here? Yeah, ADP is getting phosphorylated. All right, so that phrase, substrate level phosphorylation, they're zooming in to how does ATP get made, which comes from ADP. And so if you stick a phosphate onto ADP, you get ATP. All right, that's what you want. And what they're trying to say here is that happens at the substrate level. So now you got to go back to that figure and say, what does that mean? Okay, so stage 2, glyceraldehyde 3-phosphate down to pyruvate. And then you can see the red squiggles saying that's where you got your ATP being made. So substrate level, what that means is these molecules in the pathway, these are the substrates, and they're phosphorylated. They got the little purple P's over here. All right, and so what they're trying to show is at this step, you get the bisphosphoglycerate donating off a phosphate, the ADP, and you get ATP, and then the other product is phosphoglycerate. And then that goes through some reactions until you finally get phosphoenolpyruvate. Now that phosphate off of PEP is going to get given to ADP, get you that lovely ATP, and the other product is pyruvate. All right, substrate level. Phosphorylation is referring to the idea that we got to stick a phosphate onto ADP. That That's the goal. We want that ATP. Okay, substrate level phosphorylation is a term I want you to just memorize and know what that means because it's gonna come back. And glycolysis is an excellent example of it and maybe something that you've seen before. Stage 3. This is something our bodies don't tend to do very frequently because of the way we're designed. But microbes, many microbes will go into stage 3 all the time. So stage 3 is making the fermentation products. So you're going to start with pyruvate again. We left off there. So what's going to happen to pyruvate? A lot of different things is the short answer. You can get that pyruvate shunting into a variety of different pathways. And those are the different fermentation products. And what happens is, it really does depend where the microbe is living, what fermentation pathway it will go off into. But one of the nice things about stage 3 is that in that process, there's a couple of big things accomplished. One of them is this, NAD+ is generated. And I'll go back to that image of glycolysis. I'll show you why that's important. But NAD+ is generated during stage 3 very commonly. And that's really needed to keep stage 1 and 2 cooking along. The other thing that's going on during stage 3, and I'll come back to it, so I'm just going to say it really generally here, is that stage 3 is necessary for the microbe to stay chemically happy. Let's just put it that way. And I'll explain more about that. But what they tend to do when they do a fermentation, the stage 3, and they get a final product, that's not their goal. Like lactic acid, our bodies can do that. We can make lactic acid in fermentation if we're denied oxygen in our muscles, and then lactic acid builds up. Well, microbes can do that. Well, they don't have muscles, but they can make lactic acid as a fermentation product, but that's not their goal. That's not what they're trying to accomplish. And I'll explain what they're trying to accomplish. They'll just excrete that out and it's just waste. Same with, when I say fermentation to you, what kind of things do you think about that are used in human lives? Alcohol, right? So the yeast that they have brewing in those big copper kettles, they're not trying to make alcohol for a specific reason for them. And even though we love microbes in this class, they're not really doing it to help us either, or to make us feel good or altered or whatever. It's just a waste product to them. And so they excrete it out. And that's why you can't make beer or wine higher than a certain percentage, because it's a waste product. They're swimming around in their own waste. Sooner or later, it's going to poison them. And so once you get to a certain percentage, those yeasts go, can't deal with this toxic waste anymore, and they die. What would be a way to get a more concentrated alcohol solution? Distill it. There's the chemist. Yep, distill it. So distillation would do it. But you can't do that with just ordinary fermentation. At some point, I think with wine production, there might be up to, I want to say around 15% alcohol, and that's about it. So beyond that, the yeast just dies. So waste, it's waste. So let's go back to this image. So here's pyruvate, and then you're going into the green portion, which are the fermentation pathways. And they've got some options here. So here's lactic acid, that's an option. All the way to ethanol is another option. Here you have acetate, formate, CO2, H2, so a variety of gases and other chemicals. So when you look at this one, and let me just blow it up. Whoops. All right. So here, where you see pyruvate going to lactic acid, you see NADH to NAD+, and then they say that goes to stage 2. All right. So that goes to stage 2. So what is going on here? So there it is. NAD+ right here. So it went over to, whoops, stop it, went over to here. I suppose, oh my goodness. Drive me nuts. All right, so this NAD plus notes back, so to speak, where it's needed here in this step. Now, I'm not gonna ask you to memorize this step. I'm just trying to get you to appreciate. One of the nice things about some of these fermentation pathways is it does make NAD plus, which feeds back into the stage 2 area. And that can keep this moving. You have to have enough NAD plus to keep this moving, and we don't, us microbes, they don't really like save up NAD plus, that's not how it works. So NAD plus has been pretty low supply in those cells. So it's good to keep that up and going, yes.
Different products of the stage 3 peptides habitat.
Yes, very much. And so, and what, you know, because some of the enzymes that they have are totally dependent on what they've been used to eating, so to speak, for just the idea of natural selection and adaptation. Other questions on glycolysis? So don't freak out thinking you have to learn every single thing on here. That's not what I'm asking. Okay, but there are some key points to be thinking about, which I put in the text there under stage 1, 2, and 3. I think this might be something more like what, I don't know if it's this one or this one. These are just a study off of. So this one has some nice text in there. So I just put those in. It just depends what your brain prefers. I prefer this one. My brain likes this one better, but whatever works for you. Okay. All right. So here, pyruvate. Let's go back to pyruvate. We're going to get more into fermentation later, but let's just finish the thought here on the TCA and the electron transport chain. But okay, so let's go to pyruvate. So now what we got to think about is what can happen, what's one option that can happen to that pyruvate? And one option is this is the TCA cycle. So if you look at what's going on in the TCA cycle and say, why would a microbe do this? This phrase may sound like, so what? Pyruvate completely oxidized to CO2. Yeah, so what? Actually, this is a really good statement because anytime you can completely oxidize something to CO2, you get more bang for your buck. Okay, fermentation doesn't really allow for that. If we go back to the stage 3 of fermentation, whoops, let me go back to the one I like. So these products, lactate, ethanol, acetate, formate, a little bit of CO2, but ethanol is not CO2. Lactate is not CO2. You did not completely break down glycolysis or pyruvate. This is an incomplete breakdown. Incomplete breakdown or incomplete oxidation, not as good bang for your buck. And when I say bang for your buck, I'm thinking about ATP. How much ATP do you get out of this? So we'll talk more about fermentation in more detail, but let me go back to the TCA. So this is actually a really good thing that is completely broken down to CO2 because it creates a situation where you end up getting lovely NADH, lovely FADH2. Those are really important molecules. Explain why. And then wonderful ATP. Some microbes make GTP, which is essentially ATP. It's equivalent. All right. So the very first step, pyruvate has to enter into the TCA cycle. It's coming over from glycolysis. And it will, the first step is to go to acetyl-CoA. And then after that, you have a reaction that gives you oxaloacetic acid, OAA. And then that goes to citrate or citric acid. And then you start going into the actual cycle. So I'll show you a picture. So they call it the citric acid cycle because citrate's the first major product of the cycle part. Or the main one. All right, and then you get a bunch of reactions going. That's why I put arrow, 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 because you get citrate back to OAA. So OAA is like the stopping point, and citrate's like the starting point. It just zips around and around and around and around. As long as there's enough pyruvate coming over from glycolysis, you'll have the TCA cycle cooking along. Okay, so again, you don't have to know step by step other than the beginning steps are good to know in the sense that you have pyruvate coming in. Here's your acetyl-CoA, and then you have those two reacting, acetyl-CoA and OAA, when you get your citrate. And then you see it zooming around into the cycle. And then you see when we say pyruvate's 3 carbons, and we say pyruvate is completely oxidized, broken down into CO2. So there's, whoops, there's one, there's another, there's another. So completely broken down. And believe it or not, that's a really good thing because it provides a situation where you end up making some nice NADPH, well, NADH or NADPH, depending on the microbe, FADH2, and then GTP or ATP. So looking at this cycle, I want you to know what kicks it off, the very start of it. And then the things I circled here, that's kind of like the big deal. Like, why would a microbe do this? All right. So as long as they have the pyruvate coming in constantly, they'll keep running this cycle over and over and over. And so it gets you NADH and FADH2 are really important. I have another slide explaining a little bit more about them. And then of course we know GTP and ATP are really, really important. I also put these slides in. Whoops, what happened? From your book. So again, whatever one you like to study off of is fine. This has a lot of words on it if you want to use it for studying. All right, but when you look at it, what's really happening is pyruvate's 3 carbons. So you get 3 CO2s are produced. So we say pyruvate's completely oxidized. Then through that process, you end up creating a situation where you get NADH and FADH2. And ATP or GTP. But when you look at how ATP is getting made, it's substrate level phosphorylation. I'll go back to the picture in a minute. And then some microbes, instead of ATP, they'll make GTP, but often then they convert GTP to ATP. So we just say it's equivalent. All right. So when you look at the cycle, ATP really is getting made at the substrate level. That's what we have happening. So let's go back to that image, the cycle. So we have right here. Where you have the, well, here they're showing the GTP. Does any of the others show the ATP? Oh, yeah, I don't know. Either all of them are kind of showing the GTP, ATP. Okay, so why do we care about NADH and FADH2? So if you think about NADH, sometimes they'll make NADPH, and then the FADH2, and all of these depends who's talking, but they can be called energy carriers, electron carriers, reducing agents, or reducing molecules, electron donors. All of that means essentially the same thing, and these all qualify. So if you are a microbe, figuring out through the laws of natural selection and adaptation, figuring out a way to create NADH and NADPH or FADH2 is good because you want those to be used in other reactions where electrons are needed. So reducing agents, I tend to call them reducing agents or electron carriers because they're going to donate off electrons is what they're going to do. And so I tend to say reducing agents when you give if you are donating off an electron, you are reducing something with that electron. So that's why they say a reducing agent. It's an agent of reduction, all right? So, but essentially, their overall job is to donate electrons to some acceptor molecule. And so when you look at, you know, I don't know if the chemistry department still has that big poster displayed out in the hallway where they had all the like glycolysis, TCA, electron, and then they had all of these other secondary pathways. It's just phenomenal. So when you start thinking about everything going on in a cell and all these reactions happening at once, you're going to need electron carriers in order to shuttle around and donate electrons where it's necessary. All right, so by the time the TCA cycle is done, you think back, we started with glucose through glycolysis. All of that has been converted to CO2, which is good. That means a complete breakdown or oxidation is really good. But we're not quite done yet in terms of getting the payoff. And so we still have to think about what are the NADH and the FADH2 molecules doing? What are they doing? They're going to shift into an electron transport chain process. It's going to end up getting them a lot of ATP. We often refer to it as oxidative phosphorylation. So if you've taken animal phys, biochem, human phys, that's probably how they refer to it, which is understandable. However, in this class, I really need you to step out of the animal human world and get into the microbe world. I want you to think about it as electron transport linked phosphorylation. That's the better way to think about it in this class to limit confusion. If I say the word oxidative to you, what molecule do you often think about? Oxygen is typical, right? Oxidative phosphorylation, you're doing that right now in your mitochondria. Right? We're aerobic organisms. We use oxygen to get this to happen in our mitochondria. But we're going to learn that tons of microbes can still do an electron transport chain, but they're not going to put oxygen anywhere in it. So that's why I want you to stop thinking about oxidative phosphorylation means oxygen's involved. Get out of the human animal world, get into the microbe world. When you say oxidative phosphorylation, shift it to electron transport linked phosphorylation. And that will be a better way to think about it. All right, we'll stop there. We'll continue on Friday. Thank you for your patience. Please spend some time thinking through this before Friday.